For most of human history, bacteria were a huge threat to human survival. When people say that a cut used to kill you, it was often because of a deadly bacterial infection. The Black Death, the world's deadliest pandemic, and a less destructive but still devastating Justinian plague were caused by the bacteria Yersinia pestis. The disease tuberculosis, once called the White Death, used to kill 1 in 7 Americans. This all changed in 1928. A Scottish doctor named Alexander Fleming left out petri dishes growing bacteria. Overnight, mold grew on the dishes. In the area around the mold, bacteria were unable to grow. Fleming discovered that the mold produced a compound that was able to kill bacteria, but was harmless to humans. Although this new medicine, called penicillin, didn't take off for a few years, it became mass-produced during World War II. Penicillin was created with factory efficiency, and the race to discover new antibiotics had begun. In future years, more antibiotics were developed. One of the most important antibiotics was streptomycin, an antibiotic which took years to find and test, was successfully able to kill the tuberculosis bacteria. These antibiotics all have different mechanisms of action. Penicillin is an example of a beta-lactam, a molecule which degrades the cell walls of a bacteria faster than they can be repaired. Some antibiotics are macrolides, which target bacteria's ribosomes. Bacteria ribosomes are different from humans. 50S and 30S subunits as opposed to 60S and 40S subunits, and macrolides will bind to the bacterial subunits, preventing the bacteria from producing proteins. Bacteria have responded to antibiotics. Because bacteria have short lifespans and fast reproduction rates, natural selection can act efficiently upon them. Bacteria have evolved genes that prevent antibiotics from doing their activities. Some of these genes create pumps which pump out antibiotics. Some create enzymes which degrade these antibiotics, such as beta-lactamase, which degrades beta-lactams. In our modern day, humans often abuse antibiotics. They are used liberally on farms, often prescribed for diseases which are not caused by bacteria, and people often don't finish antibiotic treatments, allowing remaining bacteria to flourish and develop resistance. At our current rate, antibiotics are threatened with impending uselessness. This future may not be as distant as we hope. Scientists have come up with an acronym ESCAPE to identify species of bacteria which have developed resistance to multiple drugs and threaten global health. This includes Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. In the 90s, during an outbreak of Yersinia pestis, scientists isolated a multi-drug resistant bacteria from a 16-year-old boy. The bacteria had plasmids that produced beta-lactamase along with other enzymes, allowing it to neutralize ampicillin, tetracycline, streptomycin, and a slew of other antibiotics. Before this, Yersinia pestis was thought to be susceptible to all antibiotics. While it is not as high of a risk to public health as other escape pathogens, this incident reminds us that the old pandemic-causing pathogens of the past still threaten us. In response to this, scientists have had to find new methods to control bacteria. One method is to produce new antibiotics, but this method is expensive and cumbersome. A new drug costs about $1.7 billion to produce and has to undergo 4 phases of rigorous clinical trials, taking nearly a decade to complete. In response to this, scientists have looked away from the lab and to a natural source. The bacteriophage, one of the oldest life forms on the planet, has potential as a solution. Phages are viruses, but instead of infecting human cells, they infect bacterial cells. These viruses don't look much like any human viruses and more resemble a spaceship. The virus has a capsid which holds genetic material to produce more phages. It has a stem which attaches to a bacteria and injects the genetic material into the bacterium. The abilities of phages have been known for a long time, but early research on them was notoriously bad. It was the Nazi scientists and USSR scientists doing most of the early research, not Americans or other Westerners. Because of the shoddy scientists used on the other side of the Iron Curtain and nationalistic, anti-Soviet and Nazi sentiment, early phage research was ignored. Only recently, the phage's potential has received attention. An essential distinction to make is between lytic and lysogenic phages. Lytic phages enter the cell, replicate and translate their genome, then cause the bacterial cells to die, or lyse, releasing the phages into the environment. In contrast, lysogenic phages enter the cell and integrate their genome into the bacteria's genome. They then enter a period of dormancy, existing in the cell for a long time before switching into the lytic phage and killing the cell. For phage therapy, lytic phages are needed. Something that makes phage therapy quite different from antibiotic therapy is phage specificity. Antibiotics are wide in scope and kill many different species of bacteria. This gives phages an advantage over traditional antibiotics. They don't indiscriminately kill bacteria. The body depends on a diverse microbiome, and antibiotics ravage these ecosystems. But this specificity makes phages harder to work with. Phages are so specific that multiple different species are required to target an infection. Scientists need to create a phage cocktail to use in therapies. These cocktails consist of multiple different lytic phages. Balancing the size and diversity of these cocktails are important. On one hand, a cocktail which lacks diversity will fail to treat an infection, but on the other hand, a cocktail which is too diverse fails as well. Early trials show that in cocktails of too many phages start to react with each other and stop working. Another concern is possible toxic effects. One part of this is making sure that the phage particles don't adversely affect the body. The other part is the release of endotoxins. Endotoxins are toxins and bacteria which are released when bacteria die. The release of these toxins is one of the main causes of disease from bacterial infections. This means if the phages are too good and kill too fast, their effects could cause worse disease than the bacterial infection alone. A subset of phages to avoid are temperate phages. These phages shift between the lytic and lysogenic phases. These phages can be helpful when in the lytic phase, but when they enter the lysogenic phase, problems occur. Because lysogenic phages integrate into a bacteria's genome, they have a chance of becoming a vector to spread antibiotic resistance between bacteria. The immune system also causes problems. While phage therapy does not risk causing a deadly immune response, like virus-based gene therapy does, the immune system will see the phages as foreign entities and clear them out. Phages do not just have potential as a future treatment, but also as a vaccine vector. Scientists have a few methods of delivering vaccines, but a phage vector has great potential. Phages can replicate in the body using bacteria, improving efficacy. They are also able to show a dense array of antigens, the molecules that teach the immune system in vaccination, and as they get better understood, they can be very fast and easy to develop. Using phages to amplify antigens have been used for coronaviruses in response to a currently ongoing MERS outbreak. Korean scientists used phages to amplify the S2 antigen for use in antibody screening. This amplification is not vaccine production, but uses similar mechanisms. The method does work to produce antibody tests, which are important for cities to understand a pandemic. Currently, no companies are working to produce a phage-based vaccine for the SARS-CoV-2 virus, but this could be a potential future platform. Despite all the challenges of implementing phage therapy, it will likely become a treatment in the future. Beyond the basic mechanical issues of making a treatment that works, there is an economic and approval roadblock. For one, treatments need to be approved by the FDA in a 4-step process. First, there are the preclinical phases, where scientists need to find a treatment to cure disease. This also includes testing the drug in cell cultures and 2 animal models. Next, there are 3 trials in humans to test safety and efficacy. Finally, the treatment is approved by the FDA and can be manufactured. The research and development also requires significant investment. Big pharma companies need to see the drug as potentially profitable. Research takes millions of dollars, years of work, and many failures to succeed. But phage therapy has great potential to be very profitable. Biotech companies are pumping tens of millions of dollars into research. The question then is not if, but when phage therapy will be put into use.